Hi everyone!
You're listening to SciPub,
where we are talking science.
Not only for the experts, but for everyone.
Welcome to a new episode.
Hello everyone, this is SciPub. Today we are interviewing our guest, Lydvina Meister. She's a postdoc in the Neurodevelopmental Dynamics group here in PRBV. So she will answer our questions today. So hello, Lydvina.
Hello, thank you for inviting me. Thank you for coming.
This podcast is about neurobiology, as you know, is your field. So I wanted to ask you to start, what's neurobiology and what do we understand under neuroscience and neurobiology?
Okay, so neuroscience, it's a really wide field, a multidisciplinary field, and it includes many fields. And one of these fields is neurobiology. So neurobiology focuses much more on the molecular and cellular mechanisms into the neuroscience, but for example, you have other fields such as the cognitive neuroscience, the clinical neuroscience, or even the neuropharmacology that focus on the biochemistry of neurons. But all these fields focus on how our central systems works and how our brain works, and that's the main compound of all these fields.
Okay, so, and also for the people who is totally, doesn't know anything about neuroscience, can you explain a little bit what's a neuron and what's the brain form of?
So, I would say that a neuron is a cell. You have many different cell types in your body, and the neuron is one of the main compounds of your brain and of your spinal cord. So, such are the other cells. The neurons is composed by a membrane and by a nucleus that get the genetical information. But the neuron have two specific characteristics, and one of these is that the neuron has an excitable membrane. So in order to communicate with the other neuron and the surrounding environment, through the electrical signals, it can communicate with the other cells and the surrounding cells. And the second specific characteristic, that neurons have different morphology. I think you've already saw it in many books. So the neurons is kind of round with the nucleus, and then you have kind of an extensive arm that we call the axon, and at the end of the axon you have kind of fingers like protrusion that connect to other neurons, and these protrusions are called dendrites. And the dendrite of a neuron is connected to the dendrite of the next neuron, and like this you make a whole network of neurons. And in order to have a functioning brain and a brain that works, you have some support cells that we call glia cells, and these glia will secrete, for example, myelin that will isolate each neurons in order to have a good connection and for the electrical signal to transfer the information correctly.
Okay, so following on this neurobiology field, what do you think we can as a society get from studying neurobiology?
I would say that it's probably a stupid answer, but it's super important to know and to understand how brains work and how cells communicate between themselves and how they communicate with the environment, because you don't only have neurons, you also have vascular system in the head. So how these neurons connect with the vascular system, and it's important to understand in the physiological condition how these cells work. And in the second step, that will help us to understand disease, when something doesn't work, when you have a disorder, how can we work on these mechanisms to try to cure some disease such as Alzheimer or Parkinson's?
Okay, so also I guess I know this is your field, so and you are studying developmental neurobiology. I guess this also could apply to this developmental. So why do you think also the developing system is important to be known?
I think that don't know that much on how connections are made during development, how the brain is built, and that's one of the main questions of our field, neurodevelopment. How do we make connection? How these cells get connected? Why these cells connect to this one and not the other neighbors? And this is one of the main questions. Try to understand what are the mechanisms, what are the signals, what are the morphology, what are the cellular components that makes this connection and makes this brain to work as it is.
Okay, thank you.
So now that we are in the topic, as we said, you are studying developmental neurobiology. So can you explain a little bit more about your own work?
Yeah, of course. So I'm working on the hindbrain. So the hindbrain is the most posterior structure for the brain and is responsible of life-sustaining function for your everyday life, circulation, respiration, some function that without them you couldn't live. And in the development, during the development, this structure appears as what we call, as 7 metameres that we call rhombomeres. And in between these 7 rhombomeres, you have what we call the boundary. So in our lab, one of the main questions is to study this boundary. So we know that this boundary have different functions, and one of the functions that we have been able to decipher in our lab is that these boundaries works as mechanical barriers. So a cell that is in a rhombomere cannot intermingle with the other rhombomere. They are blocked because of this boundary. And there is another thing that we have been able to see in the few last years, that while the brain is undergoing neurogenesis, that means that all the brain let's think that at the beginning all your brain are stem cells. Mm-hmm. And these stem cells we slowly specify to neurons. And while all the cells of rhombomere are specifying to neurons, these boundaries stays as stem cells. And we try to understand why do they stay as stem cells. We think that these are a pool of cells that we slowly refine the final proportion of the cell population. But in my project, I want to see if there is any morphological differences between this boundary and the rhombomere. For example, the cell cytoskeleton, does these cells have a specific cell compounds, cell cytoskeleton to keep them as stem cells? So that's the main question of my project.
Super interesting. I know that you're working with zebrafish, so could you explain us what zebrafish is for our audience and why is it useful to use it in research and specifically in your research?
Yes. Zebrafish is a vertebrate, so we are exactly in the same clade as humans, so we can compare zebrafish with humans. And in fact, the hindbrain, the structure which I'm working on, is the most conserved vesicle along evolution. So working on the mechanisms in the zebrafish could help us to understand how it works in humans. But there are many advantages. For example, zebrafish has external fecundation, and so the male lay his sperm and the female lays her eggs, and then you have external fecundation, so you can easily work from one cell stage. And what we usually do in the lab, that from one cell stage we undergo some injections, so we can modify at one cell stage the DNA of our zebrafish. The brain of the zebrafish is totally transparent. And for example, we want to say, I want to follow this cell population in the hindbrain or in other part of the brain. So I will modify my DNA in order to be able to put red fluorophore under the control of the gene. So I will be able to follow this cell population. You have to put the zebrafish under the microscope, and then you can follow every cell population using fluorophore or whatever other molecular techniques. And you put your embryo under the confocal, under your microscope, and you can easily follow your neurons, cell population, cell behavior under the confocal. So that's actually a really important thing in our field, be able to see how it works. And using zebrafish, we can easily follow our neurons and our cell population and our mechanisms, and I can look on the cytoskeleton, and it's really easy.
Okay, and do you think that the research that you are doing will be then applied to study or to gain knowledge in some kind of a disease?
So first of all, I think that just understanding how our brains work is one of the main focus that I have. But of course, I hope that some days that could help in some therapeutics. That we could, I mean, it's gonna be a really, really, really preliminary work, to be sure, but I hope that if we know how cytoskeleton is arranged in our brain stem cells that we call neuronal precursors, be able to extract some and take some cells from patients, induce them, and just by looking on the apical cell surface, so on your cytoskeleton arrangement, be able to drive these cells toward a specific cell population, that will be amazing. But from the preliminary work that I'm doing, I'm really not sure that in a few 10 years we will be able to create therapeutics. But our focus, I would say, in academic science is just understand how our brain works, and from zebrafish try to see how it works in mice, since I mean it's not an external fecundation, so we have to open the mice, and then from mice maybe try to see if it works and if the mechanisms are similar, and maybe go to humans. But this is one step after the other.
So maybe your work can be applied to neuroplasticity. Can you explain what neuroplasticity is?
Okay, so neuroplasticity is the ability of the network to get rearranged, to have a reorganization in order to have a better growth. For example, I have an example in mind. A person that gets blind at some point of his life, he slowly will have the reorganization of the neural network, and his sensory input will change, and this person will slowly get better better sensory abilities. He will get better taste, better hearing. So this an easy example to understand what we say by neuroplasticity.
Okay, thank you.
Okay, so now we also went to the street and asked people in the street what they thought about neuroscience and neurobiology, and if they had any questions for you. And a lot of people ask, like, how do we know the brain? How much do we know the brain? And do you think that we will ever be able to know completely this structure and the development of it?
Okay, I would say that brain is a really complex structure, and there is a big sentence in our world saying that we just know 10% of our brain. I don't know if it's true or false, but for sure we do not know anything about we know a really, really small proportion comparing to what we could know about the brain. I think there is 100 billion of neurons, 10 million of billion of connections, so for sure we have much more to know. And we have still many in all science, in all neuroscience fields, in cognitive science, working on the sense, in neurobiology, in neuroscience. We have, in all the fields of neuroscience, we have much more to know. But what do we know from 100%? I really don't know.
Okay, yeah, probably there's still a lot of work, and for sure we won't see the whole structure.
Never. But I think that with the new technology, such as the molecular tools or the new technology like live imaging, and with the new microscope that we are building, these go very, very fast.
Yeah.
Yeah, that's true.
So also another question that was asked by the audience is, what's your day like? Because a lot of people didn't know what a neuroscientist is doing in their day. So can you explain a little bit your day?
I think one of the main advantage of our work is you don't have 2 days that are the same. Every day is different. So you don't have 2 weeks that are the same. So I would say that in neuroscience, and at least working on zebrafish first, and this is kind of the routine that we have. So we get our material, so we get our fish, we then do the experiment. Our experiments are really different. We're sometimes working with the RNA, sometimes we are working with antibodies, so with protein. This depends. Some experiments are 2 days long, some experiments are 3-5 days long. Then you go to the microscope, you put your embryo under the microscope or whatever, you do your experiment, and then when you get the results, you analyze your results, and regarding your results and your hypothesis, you always create new experiments regarding the ones before. So I don't have a schedule. One of the positive and at the same time negative things working with zebrafish that you follow the development of your brain structure, and the zebrafish. We are quite really lucky about that, since the zebrafish development is really quick. So in 3 days, the brain is almost all specified. So you have something that looks like really an adult brain. So you think alone, and then every 2 weeks have a lab meeting all together. We show our results, and the good thing with that is that we think all together. Sometimes you have lab mates that see things different than you, so it's really nice to have different opinion about your work. And I would say like once a year, you go to a worldwide congress, so you can meet the community that are working on the same field, or I would say related field, and you show your results. And even sometimes you get some collaboration with people in other parts of the world. So that, I would say, one of the advantages of our work as a scientist.
So now we want to ask you more personal questions. So now you're a postdoc student in the UPF. So could you tell us what is your background, what's your path, how did you reach this point?
So in fact, I'm French, so I did most of my studies in France. So I did my bachelor in a small city next to Paris. I did a bachelor in health science, quite general field, and then I really like working on genes and modifying genes and thinking about genetics. So I did a master in what we call gene, cell, and development. So I focus my career on working on genetics. And then after this master, I did a PhD in a small village next to Perpignan that we call Bagnols-sur-Mer. And it's in the south of France, and I've been working on the evolution of our head muscles. Actually, I've been working on a small animal that we call Amphioxus, that is 6 centimeters long. And I've been working on the head muscles because our head muscles are really different from the rest of our body. And the head muscle of this small guy are the same in all the body. So I've been working on the mechanisms of creating the mechanisms of head muscles in this animal, comparing with of the knowledge of humans, and try to see how our head muscles are built. And this field that we call EVO, that is a new field, mixing evolution and development. And then I moved to the UPF. I wanted to focus on development and on neurobiology and neurodevelopment, and that's why I'm here.
Super nice. And would you say it has been like easy path or more with ups and downs?
Without any doubts, with ups and downs, and even much more downs than up. I think that, on the contrary to the lab work that we are doing in master everything works always. When you do a PCR, it works. When you do an immuno, it works. However, in the academic science, nothing never works. So it's, I would say, 1,000 downs, but when you have an up, you forget all your downs. So I would say that keeps you awake, and that keeps you following the academic path, that you forget all your downs and you just focus on your ups. And sometimes when you have enough ups, you just put in a paper and that's your scientific contribution. But yeah, it's always downs.
And I mean, I don't want to stress you out or anything, but do you have any future plans or?
One thing after the other. So I got a fellowship with this Utopia fellowship in the UPF, that is a part of the Marie Curie fellowship. So I've just started, so I have 2 years to try to answer my hypothesis that I told before. So after this, we will see. I would say that I'm looking for opportunities, and for the moment I want to finish these 2 years, and after all I really like communication, so why not doing some exhibition, creating exhibition, or why not helping on another side scientists working in a private company and try to make some molecular tools for many scientists, whether in a private company or in a public. I really like neurobiology, so that's one of the things that I want to continue with, but I don't know what's the future, and we will see.
That's good.
So now coming to the end of the podcast, do you have any advice for younger scientists or people in general?
I would say that science is really wide, and you don't have to be a really good researcher. Research in academic is really wide also. You can be if you like to be doing experiments, you can be a technician. If you really like, for example, microscopy, you can be an amazing engineer. And if you like to have big projects, you can be a researcher. But I think that the only thing that you have to keep in mind when you do science is you have to be curious. Just if you was the child that in the car with your parents, you was always saying, why this is like that? Why this happens? I think, and if you continue to think, why I have this result. You have to be always curious, and when you are curious and you always to want more, I think it's enough to do research.
And any final words or a take-home message for our audience?
I think it's a hard path if you want to be a researcher, but if you like what you're doing, just follow what your thoughts. Mm-hmm. And if you come back from the lab and you're always thinking of your experiments and you go to your bed and still thinking, you wake up. If you have your career and you know that you want to do research, just follow your thought and go straight.
That's nice.
That's nice. Thank you, Lydvina,
you.
for answering all of our questions today. And it was very interesting talking to you and hearing about like how you are working here in a biomedical research park, but you've worked with so different animals and species. I think this gave us like very different insights from all other guests. So thank you for that.
Thank you for inviting me again. Thank you very much.